Yesterday we were talking about gene therapy method. So gene therapy nothing but the replacement of defective genes with the corrective genes. One of the treatment method, just like physiotherapy, orotherapy, etc. So one example is given in the book. That was the first disease that was treated through gene therapy method. Now the first clinical gene therapy was given in 1990 by French Anderson to a 4-year-old girl who was normally suffering with adenosine deaminase deficiency, normally called as ADA, adenosine deaminase deficiency. Now this disease is an autosomal recessive gene disorder because the normal gene which produces an enzyme what's called adenosine deaminase undergone mutation to form a recessive gene. So that was happening. The enzyme production is stopped. And what is the role of this enzyme? This enzyme is responsible for the proliferation of T lymphocytes indirectly because of accumulation of what is called DATP due to the deficiency of this enzyme. Another enzyme, RR enzyme, indirectly involved that affects the proliferation of T lymphocytes. Let us take simply the role of adenosine deaminase enzyme is to proliferate the T lymphocytes. Okay, if this enzyme is absent, we have received only the non-functional T lymphocytes. They are unable to elicit immune response against the invading pathogen, so that the disease should be treated. So, what is that one? Adenosine deaminase deficiency disease. This disease is normally called as SID. The expansion for that one, severe combined immunodeficiency disease. Severe combined immunodeficiency disease. So it's a group of disease. One such disease is adenosine deaminase deficiency, simply called as ADA. Now, for the treatment of the disease, there are different methods that are adapted. I'll explain later now. One such method, the recent method, is gene therapy, replacing the defective genes with the corrective genes. And here, what are they doing? Now, the lymphocytes from the affected person or from the patient's blood are collected and they are cultured in a nutrient medium. Then, the gene is inserted. Actually, the cells are taken out because we are culturing the cells in a nutrient medium after taking out from the blood. Then the gene for the disease, the gene for the production, not the disease, for the production of that enzyme, sorry, has to be inserted into the cells so that the cells can synthesize the enzyme, so they are effective now, can fight against the invading pathogens. Okay, so there are 3 ways by which we can cure the disease. This is what is called SID disease, severe combined immunodeficiency disease. Can be treated by 3 different methods, either by means of bone marrow transplantation or enzyme replacement therapy or by means of what is called gene therapy. So I will take this one, the gene therapy method, then we will go for that one. So what are they doing actually in that process? Normally, the lymphocytes, the defective lymphocytes from the patient's blood are collected and they are cultured in a nutrient medium. Then, the gene actually coding for the enzyme, the gene coding for the enzymes, what is called actually cDNA. This is called ADA cDNA. This is the gene, ADA gene. Now we are taking that one in the form of ADA gene, cDNA gene, ADA. So adenosine deaminase enzyme gene. Now this is what's called cDNA. The gene is in the form of cDNA is inserted into the cell to produce an enzyme. What is cDNA? It is a complementary DNA. I'll tell you more about that one. A complementary DNA is the one which is being synthesized By reverse transcription method, by using what we have, the ribosomal RNA, or we can use the messenger RNA. By using the messenger RNA, a complementary strand of DNA has been produced. That DNA is called complementary DNA. It is being actually produced by means of reverse transcription process. I will explain later about this one once more. Okay, now this is ADA-CDA gene. So the cells are taken out and cultured in a native medium. Then the gene is inserted or introduced into the lymphocytes with the help of a vector. The vector used for the transfer of genes is nothing but retrovirus. The vector used for this one, retrovirus. This retrovirus is a harmful virus. So it contains some genes which cause disease. So it is an effective vector for animal gene transfer. In the case of human gene transfer, the vectors used are either retrovirus or adenovirus. Most effective vectors for gene transfer in animals. And the retrovirus, the genes which are harmful in that virus are spliced out, removed, so that we can make that virus genetically disabled, unable to cause the disease. Now it is used as a vector. After splicing out the harmful genes, now the retrovirus is used as a vector, and now it is called genetically disabled retrovirus. Meaning for that one, it doesn't cause disease, the disabled retrovirus. Now we have to take the gene ADA gene. The ADA gene is to be cloned with the help of bacterium. So cloning is performed with the help of bacterium, with the help of plasmid, and the transfer is affected by retrovirus. So this is a vector for transfer, that is a virus, and the bacterium, the plasmid is used as actually vector as well as a cloning vehicle for multiplication. Now this is the bacterium carrying plasmid with the cloned normal human ADA gene. So for cloning, for production of many genes, we are using the bacterium. Now this is a bacterium that is carrying plasmid. That plasmid has actually what's called normal human ADA gene. Okay, it is extracted from a normal cell. Now from that one, what is happening? The gene has been transferred to this disabled retrovirus. Now the retrovirus is highly infective. When it is allowed to just incubate with the cells, it infects the cells. So now this cloned ADA gene, because of production of many copies, is incorporated into virus. So it is incorporated virus from the plasmids for cloning, production of many copies. It is being incorporated into the virus. This is the process. Cloned ADA gene is incorporated into virus. Then the virus is allowed to infect. It is incubated with the what is called the cells. The T cells with the disabled ADA gene isolated from sick patient. So it is being incubated with this virus. So that what is happening? The retrovirus infects the T cells, transfer ADA gene into cells. So this is the T cells. Disabled means not having functional ADA gene, so that there is no functional enzyme, ADA enzyme, adenosine deaminase enzyme. Now when the virus is allowed to incubate with these cells, they transfer actually what we call ADA gene into cells. These are all the disabled cells. Now the genes are transferred here. I see that on the black lines. Then what will happen after transferring the gene? These are called the genetically engineered cells. Genetically engineered cells. Now the cells are grown in culture to ensure whether the ADA gene is active or not. Culture medium. In culture medium to find out whether the gene is active or not. So this is a genetically engineered, what's called the cells. We can see genetically modified or genetically engineered cells having the gene for the production of ADA enzyme. Okay, now all these processes have been done outside the body. After the cells have been genetically engineered, the cells are once again re-implanted into the body of the child, so that now the gene is functional, able to produce the enzyme, That enzyme is responsible for the proliferation of the T lymphocytes that makes the child effective against the invading pathogens. This is the thing what is happening. Now, it is also called bubble boy syndrome. Bubble boy syndrome. The disease is also called bubble boy syndrome. The disease is also called bubble boy syndrome. The meaning for bubble here referring to a small incubator to keep the child in that incubator for some time against infection, to prevent infection. That's why it's called bubble boy syndrome. It was noticed a boy was kept in a sterile bubble, referring to a small incubator carrying only one individual at a time. So this is the mechanism what we have. So retrovirus is used as a vector. The bacterial plasmid is used as a vector for cloning the gene. The retrovirus is used to transfer what is called the gene into the human cells, the disabled T lymphocytes without ADA gene. So this is an illustration. I will give more information how far actually the disease is being cured by different methods. Okay, let's see. So the first clinical gene therapy was adapted in the case of a disease, a patient suffering from adenosine deaminase deficiency enzyme. It's one of the actually combined diseases of what is called immunodeficiency. It's very severe, hence called severe combined immunodeficiency disease, SID. Now this is a disease caused by an autosomal recessive gene. So the gene for the synthesis of the enzyme adenosine deaminase is found in chromosome number 20. When this gene is defective, the enzyme will not be produced. The enzyme ADA enzyme is responsible for the proliferation of T lymphocytes. Only when the enzyme is present, functional T lymphocytes are produced. Otherwise, there is no functional T lymphocytes produced. Okay, now, what's the reason for that one? When there is a deletion or dysfunction of the gene coding for ADA enzyme, that's the main cause. Deletion or dysfunction of a gene found in chromosome number 20, that one is coding for enzyme adenosine deaminase enzyme. That results in the disease. So as a result, what is happening? In these patients, we have non-functional T lymphocytes. They are actually the defense mechanism of the body. So the T lymphocytes cannot elicit immune response against the invading pathogen because they are non-functional, because of the absence of enzyme. So the right approach for the treatment of the disease, namely the SID, it would be normally to give functioning ADA, that is nothing but functional adenosine deaminase enzyme. What for? To break down biological products. What products? Toxic biological substances or the products. So I mentioned this ADA enzyme is responsible for the proliferation of T lymphocytes. So without which the proliferation of T lymphocytes is less. So the least count of T lymphocytes make the person susceptible to infection. Even a small infection cannot be tolerated by that individual. The disease is also called bubble boy syndrome. Bubble boy syndrome. Because the infected child is kept in a sterile bubble. A bubble is nothing but a small incubator, can carry only one individual at a time. Okay, so what are the treatment methods? How would you cure this disease, adenosine deaminase deficiency? So we have three different methods. Treatment for just SID or ADA deficiency. It could be cured, number one, bone marrow transplantation. Because bone marrow is a place of production of the lymphocytes. We have the stem cells found in the bone marrow for the production of lymphocytes. That's why we have to replace it. Let's see now. Then enzyme replacement therapy. So the enzyme adenosine deaminase enzyme, that is to be replaced if it is defective. The last one, the current method what we have, the recent method adapted, gene therapy. That is replacing the defective ADA gene with the normal corrective gene. That is called gene therapy. Now let's see these three methods, at least one or two statements for the treatment. The possible methods of curing SIB or ADA deficiency are, we have temporary methods as well as permanent methods. Now the temporary methods for a short term. 1. Bone marrow transplantation. Out of the 3 methods, this is a temporary method. So the defective immune cells, we have the cells produced by the bone marrow. So there you have the defective cells are present, the one which produce what we have AD enzyme. So the defective immune cells are replaced with healthy functional immune cells from a donor, the one who donates the bone marrow. So it is only a transplantation from bone marrow from a healthy individual to a defective individual to substitute a defective bone marrow. The second one, we can directly just replace the defective enzyme, and that is called enzyme replacement therapy. Enzyme replacement therapy. So in which what is happening? The functional AD enzyme extracted from another individual is injected into the patient's body so that it can work for a short period, not much long time. So these are all the temporary methods. But the advanced method, what we call the gene therapy. The defective ADA gene has been replaced by a normal healthy gene. Now what are the steps involved? First we have to remove the lymphocytes from the blood because they are the main producers of what we have the defense mechanism. So step number 1, the lymphocytes from the blood of a patient is removed and grown in a cultured medium which contains nutrients. The second one, a healthy, just what is called a functional human gene. This gene is called ADA-CDN. ADA-CDN. What is cDNA? This is what's called complementary DNA. cDNA means complementary DNA. So I'm writing here cDNA, the expansion for that one, complementary DNA. It's called complementary because the bases are complementary to what is called mRNA. Now, from the mRNA, suppose you are taking this mRNA, then through what is called reverse transcription method, reverse transcription, we already studied what I mean by that one, with the help of reverse transcriptase enzyme. Now we are getting what is called DNA. This is a DNA single strand. This DNA is called complementary DNA. So the complementary DNA is nothing but a copy of mRNA, which is produced by reverse transcription method. The enzyme reverse transcription, reverse transcriptase. So the DNA which is produced by reverse transcription of mRNA is called complementary DNA. Now the cDNA, there is the gene for ADA, that is called DNA, cDNA, ADA-cDNA. The gene is called AD, adenosine deaminase. The gene is extracted by using what's called reverse transcription process with the help of mRNA. That is called cDNA, that gene. So that gene is introduced to the T lymphocytes. The major vector for transferring the gene in the case of animal cells are number 1, retrovirus. This is acting as a vector, retrovirus. Or we can use adenovirus. These are the 2 major vectors used for the transfer of genes in the case of animals. So a healthy and functional human gene for the synthesis of ADA, adenosine deaminase enzyme, has to be introduced in the T lymphocytes, the defective T lymphocytes using a retrovirus. Now all being done outside the body of the patient. The cells which are formed as a result of this transfer are called genetically engineered cells. So the genetically engineered cells are subsequently returned back to the patient's body. These are the 3 major steps: isolation and culture, then introduction of the gene that is into the cells, and finally introduction of the genetically engineered cells into the patient's body. Okay, these are all the steps. But normally the genetically engineered cells are not immortal. Not immortal. So immortal means can survive for a long time, not immortal. They can die. So the patient requires periodic infusion of genetically engineered T lymphocytes. So he cannot survive for a long time with one what is called transfusion or infusion of the gene. He needs periodically the infusion of what is called the genetically engineered cells again and again. So it is not a permanent cure. So the permanent cure for this disease would be if ADA gene actually from the bone marrow cells are transferred into the early cells of the embryonic stage. Here what we are doing in adult, but it should be done in the case of embryonic cells. The embryonic cells become what is called stem cells, producing more and more T lymphocytes, so they can produce ADA enzyme. So the permanent method is transferring the gene obtained from the bone marrow cells into the early cells of embryonic condition. There is a permanent cure for this one, but the cost is very high. It cannot be done easily. That's why it was conducted only once in the case of a child. And the name of the child, Ashanti de Silva. The name of the child, it was conducted Ashanti de Silva. Okay, that's about what we have, that is curing methods for SID or ADA deficiency. Some related statements regarding SID or ADA deficiency. Now, the first clinical gene therapy normally was given in 1990 by French Anderson to this disease, what is called the SID, to a 4-year-old girl called Ashanti de Silva with adenosine deaminase deficiency. It's also called by name severe combined immunodeficiency. So adenosine deaminase enzyme, what's its function I mentioned already, responsible for the proliferation of T lymphocytes. The defective ADA gene is present in chromosome number 20. The affected individual is kept in a sterile bubble. That's why it's called as bubble boy syndrome, another name for SID. Okay, the bubble is nothing but a small sterile incubator keeping only one individual at a time. Because such individuals are highly infective, they have to be kept in separate incubators. Such a small incubator is called bubble. Okay, with this I concluded what we got the gene therapy. We have to pass on to the next one, stem cell therapy. We'll do it in the next class.